So thank you, Sarah, for giving me the opportunity to talk here at this Medicines Discovery Catapult Connect webinar about the hit identification and why understanding your target is key. So just a little bit about DomainX for those who don't know. We're a contract research organisation based within the sort of southwest of Cambridge, a biosciences hub there. We're around about 50 to 60 scientists, and we've got just over 2,000 square metres of lab space. And it's an all-in-one facility, so we've got chemistry and biology under one roof. Our clients are widely spread between big pharma, agrochemical, small, medium, and large biotech, startups, virtual companies, as well as major academic institutes. And over the years, we've developed compounds that got into clinical candidates, preclinical development. We're cited as inventors on a number of different patents, and obviously authors on lots of different peer-reviewed papers. And we are a dedicated CRO. We've got no internal research projects. And critically, all of our staff are on one site. And that's key to this talk, is that we work incredibly closely together. As I said, we've got a comprehensive medicines research platform. So that means we've got chemistry, protein science, and assay biology all together on one site. And we cover all aspects, as you'd expect, of these divisions. So we've got computer-aided drug design, medicinal chemistry, analytical chemistry, protein expression, combinatorial domain hunting, which is particularly useful for proteins that are difficult to express, X-ray crystallography, and then within assay biology, you've got cell biology, which means we can develop bespoke phenotypic assays, biophysics, as well as biochemistry. And the teams work really, really closely together, and this is exemplified at how we go about the hit identification at DomainX. Each group isn't just siloed in its own area, so the assay biologists don't go off on their own and design a hit cascade for the target. We put the target at the centre of the project, and then we're surrounded by the experts from each team, so that's chemistry, protein, assay biology, all focus on the target and the target profile to put a plan in place. Not only just identifies a hit that's got the best chance of acting on that drug, but they've got the best chance of turning the hit into and translating that into a drug that's going to go into the clinic. For example, proteins that have different domains and different regions, chemistry can work out whether or not these different domains or regions are druggable, which are more druggable than the others, do different regions infer selectivity versus other family members. And obviously this will feed into protein sciences. Do we want to produce full length protein? Do we want to produce truncated protein? And again, this will feed into the assay biology. Can we identify where the hits are binding? And do specific regions of the proteins have different downstream effects? And are we able to design a screening cascade where these different effects can be measured? And I said, all of this is focused on identifying a hit that's got the right chemical properties, not just to act as a tool drug, but to become a drug getting in towards the clinic. In terms of compound libraries, at DomainX that we've got a virtual screening platform, so that's Lead Builder, and a fragment screening platform, all of which can feed into a successful medicinal chemistry program. And successful projects need good quality starting points. To generate those, you need good quality compounds and good quality hit matter. You don't want to run screens with large numbers of artifacts or large numbers of compounds that make poor chemical starting points, because doing this means that the compounds that are good can get lost in the noise. So in terms of our virtual screening platform, obviously this can utilize structural information or homology modeling, and we tend to screen up to about 1,000 compounds in this. And at the bottom left, you can see kind of a visual representation of some of the virtual libraries we've got. And we've got a CATS library, which are all the commercially available compounds. This isn't just a virtual library that doesn't actually exist in reality. This is a library that genuinely does exist, that means that we can purchase from them, and that enables rapid hit expansion. We've also got a highly curated chemical library called the NICE library, which is a number of interesting chemical entities. As I said, this is a highly curated library. It's QC'd about once a quarter. And this is our lead-like library. So We've got in-house internal filters, and this filters out all frequent hitters, reactive motifs, pains, and delivers Lipinski-like lead compounds. The fragment set, as you'd imagine, it's a very highly curated fragment library, diverse library, about 1,300 fragments. These are all rule of 3 compliant, and they contain a large number of 3D fragments as well as 2D fragments. We've confirmed all of their solubility experimentally at 1 millimolar, so we know that they're soluble at the concentration ranges that will be screened. And again, these are regularly QC'd and stored in nitrogen pods. And the idea of all of this is that the hits that come out of the screens are good quality compounds that are good chemical starting points. And again, it's not just about identifying a hit, but increasing the chance of actual success of the project and delivering it through into a drug. In terms of protein production, the key really is to focus on the target product profile that you want. Where do you want your compounds to bind? If you're agnostic about that, it might be better to produce full-length protein. However, it's important to produce the right and relevant protein, not just simply protein production that gives you the biggest yield. Critical to good quality protein production is a high quality QC protein. So just a single band of an agarose gel doesn't necessarily mean you've got a homogeneous protein. It's important to take it through multiple step chromatography. For example, separating phosphorylation variants by ion exchange chromatography. And again, here at DomainX, if there's ligands available, we'll regularly QC our protein by thermal shift DSF assays. Finally, in terms of the screening assay, this again, it's critically important to have good quality screening assays. And it's important to be able to assess the target in line, it's important that the screening assay you choose is in line with your target. So the technology that we use at DomainX regularly is microscale thermophoresis. And this is a technology that I've been appreciative of for years beforehand, before joining the organization. And it's got a number of advantages that we feel above other biophysical methods. So it uses very, very low protein samples. So typically about 5 to 15 nanomolar concentration in about 5 microliters. This means that you can focus on the right protein. So even if your prep produces relatively low yield, but it's in the right conformation, this is something you can use. So you're not having to move into a preparation that just produces bulk protein if it's not the right form of protein. It's also a solution-based system, and this means that the target can be delivered in the appropriate way, not just in the way that makes the method work. It can also use multiple different labeling strategies, and often here these are directed by the structural information that we can gather about the target. And this means that we're able to deliver the protein in its near physiological state, which gives us the opportunity to be able to identify hits in a method that means it will more likely translate into a drug at the end of the project. At DomainX, we've got the supporting capabilities. So that means that we have SPR, ITC, NMR, and DSF, which obviously gives us the opportunity to put the hits through a cascade. So what I'd finally like to do is end by having a few slides just on a case study, and this is G9A, which is a lysine methyl transferase, which is involved in epigenetic regulation by covalent modification of histones. And this was a target that we wanted as an oncology target. So the only available assays for G9A were alpha screens, and these weren't suitable for high concentration fragment screening. What we also wanted to do was target the peptide binding site to gain specificity over the SAM binding domain. And what we wanted to be able to do was this we felt would be able to infer specificity over other lysine methyl transferases. What this meant was that we ended up with a complicated biophysical system because it was a 3-component system. And here with this project, we ran both a virtual screen as well as a fragment screen in parallel, and this is a way of screening that we find particularly beneficial. Here's the primary hit data, and this is just an example of how the hits come out of the monolith system, the MST system. And it also highlights another advantage that we find from the system, in that actually, because of the way it runs, you get false positive detection occurring straight away. So on the top right-hand side, you can see an example of protein aggregation due to the compound. And then below, you've got ligand-induced photobleaching and then autofluorescent. And what this means is that when you get data out of the monolith, you can then focus on the good quality hits that come out and actually ignore it, discount some of the compounds that are coming out from the noise. Going on from this project, we were able to identify high quality binding hits with really good ligand efficiency. These compounds were all were confirmed by NMR, STD NMR, and a number of those demonstrated positive binding. And you can see on the right that we were able to generate a number of X-ray crystal structures for these compounds. This led a successful medicinal chemistry program, improving these compounds and was keeping the ligand efficiency incredibly high. So in summary, integrated drug discovery approach, it's essential for hit ID. So having really good contacts and good communication between the chemistry teams, protein production, as well as assay biology. The most important thing is that you're not really just identifying a hit compound, but you're identifying the starting point for the drug. So the quality of the hit compound matters highly. The better the compound, the more translatable it is. It means you can more likely to have success through your lead optimization programs into delivering a drug for the drug candidate. And it relies on appropriate protein delivery, high quality compound files, as well as a sensitive screening assay that's essential for the library that you're trying to screen. And here's a final summary slide, which just highlights the services that DomainX offer. So it's an integrated drug discovery service, and we've got a really good track record, not only of delivering high quality hit matter, but high quality hit matter that delivers into preclinical candidates and on into the clinic. I will now go through the Q&A sections and I'll see whether or not we have any questions that I can answer. What are the biggest difficulties in compiling and maintaining the compound fragment libraries? So for us, I think what we've done is the physical library enables us to, because it's not enormous, it enables us to maintain it very, very well. It means that we're able to curate it highly. I think in terms of the difficulties, it would simply be in terms of scale and having a large, the bigger the library, the more difficult it is to curate. And the first question said, what experience do all proteins work using MST as an assay platform? What percentage success rate? So DomainX specifically brought in the MST, I think about 6 years ago. So it was, we were very, very early adopters. And at the moment, I don't believe we've had any failure to develop an assay, a sensitive assay for MST. We go through quite a thorough assay dev program for our MST. So for example, we'll run a buffer screen through it, which is particularly useful. And the biggest problem with most of them is needing a good quality tool compound. And I think that's what's always the challenge, is when you have projects that come in where you don't necessarily have good quality tools or any tools whatsoever, that becomes more challenging. But so far, we've managed to develop assays and hits for, if not all, very nearly every single MST assay that's come in.